be our kind of fate walks. And the thing I love about prokaryotic cells is you have something called this run-tumble movement. All right? And basically what we're seeing here is if this is the target of the prokaryotic cell, and this is the start point, you can see that the path that that prokaryotic cell takes is not a straight, even path, right? And the reason that this is occurring, because your flagella, well, not your flagella, the prokaryotic cell's flagella spins in a counterclockwise motion. Right? I should have brought mine back, right? Spins in a counterclockwise motion. And eventually, though, it hits a point where it can't spin anymore. What do you think it does? It unwinds, right? So it essentially spins the other way, right? It unwinds. So I used to have my little satchel. I don't want to be like man purse, but satchel sounds better, right? And I could spin it, I could hold it by a strap, and I could spin it, and eventually it gets too tight. I stop spinning it, and it unwinds, right? So what we see in prokaryotic cells is while the flagella is spinning in that counterclockwise motion, it is moving towards its target. So we call that the run. Okay? But eventually it can't spin anymore, and it unwinds. And during that unwinding process, we have chaos, right? And so we call that the tumble, right? So here it's running towards its target, but now it's spun, right? As far as it can go, it unwinds, and then it tumbles, right? And so you can see that's why this movement looks kind of chaotic and jagged. Much like, get ready for this biblical drop, right? Our walk with Christ, right? Here's our end goal. Here's the path that we choose to take. A very bold approach to success, right? But, right, through faith and a lot of grace, we do eventually make it, right? And these prokaryotic cells eventually make it to their target. Now, one of the things we did, I didn't mention, but you guys are probably a little bit familiar with this, right? This idea of chemotaxis, and this is moving towards in this case, we're talking about prokaryotic cells, a nutrient gradient, perhaps. One of the things we don't really mention is this can go the other way, though, right? Prokaryotic cells can actually use this to move away from a dangerous situation also, right? So it can kind of go either way, right? But the general principle, right, we're talking about a nutrient source is, right, it's moving, right? Here the nutrient concentration is lower, it's moving towards this higher nutrient concentration, right? And so you have this kind of run tumble. Now, what we do know is that in the absence of a nutrient source, right, flagella are still spinning, and I mean, it's even more crazy, but it's run tumble, right? There's just almost kind of no rhyme or reason to it, it looks like. However, when a nutrient source is detected, then you do get, right, a little more kind of self-controlled movement towards that source. Okay? And I don't expect you to memorize this, but one of the things that I did kind of want you to appreciate is, the basic physiologic principle of how this is happening, right? And so within a prokaryotic cell, right, in the cytoplasmic membrane, so that's what we have here. So again, this is another gram-negative bacteria, but gram-positive, same basic principle, right? You have receptors for different nutrients, right? So here you have some amino acids, here you have some sugars, here you have something like oxygen. I can't remember if I'm going to have you do it this unit or next unit, but we have a paper where you guys, you read about bacteria's love of iron, right? So you could have a receptor for iron. And so, right, when a bacteria is in an environment and one of those nutrients hits one of those receptors, and that's kind of like a ding, 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 right? Something in the area is useful. So there's something there that we want. And so when that nutrient binds to this receptor, right, it starts, right, a process that leads to the flagella kind of amping up and getting ready to move you towards that nutrient source. All right, so there are receptors embedded in the cytoplasmic membrane of prokaryotic cells that are on the lookout, right, for those nutrients that they want to help them move towards that target. Okay? Oh, yes. All right. So, we talked about capsules being important for adherence. You also have something called pili, right? And pili are allowing adherence to specific surfaces. So if you talk about a pili that's allowing adherence to a very specific surface, you call them the fimbriae. What do you want? Forgive me. I assume it's Caitlin's question. She's just ready to talk.
Can only bacteria with flagella do chemotaxis?
I haven't looked at bacteria as much without flagella. I would say the answer is actually no, because what we see bacteria, well, and the reason I'm saying no is because I know in fungi, right? I don't know if you remember Professor Perry telling you they're actually non-motile, but what's really cool for those hyphae, if you remember those, right? She showed you hyphae that were kind of had septums in them, right? They're kind of segmented. And so what's really cool about flagella is that, or not flagella, fungi, right, with their hyphae, is they also kind of have a chemotaxis thing where they divide, but they divide towards the nutrient source, all right? And so I actually haven't looked at bacteria, but I'm like, if fungi can do that, then bacteria can do that, I would assume also. So I would have to double check that, but I would be willing to probably bet Alyssa's life that my evidence is very low. No, I'm going to say yes, I can double check, but again, if fungi, hyphae, they can divide it towards the nutrient source, bacteria can certainly divide towards the nutrient source, right? And what you actually see here even, right, with fimbriae, so this kind of answers your question a little bit too, this allows some movement across solid surfaces, right, so the absence of flagella. Now again, this is very slow, so if I took bacteria with fimbriae and put it on the edge of the desk next to Morgan, it's not going to be all the way down to Rachel by the end of class, but it does allow a minimal amount of movement too. Right, but that's a really good question. I'm going to say yes because of cell division, we can divide towards. All right. Fimbriae and pili are the same thing, right? Fimbriae would just be a pili that allows attachment to a very specific surface. And right, these are important components of pathogenesis. So this is going to be right one of the mechanisms by which some bacteria actually bind to a specific receptor of their target cell. All right. So remember, if we talk about pathogenesis from a bacterial standpoint or a viral standpoint, I think we've already kind of mentioned this in some form or fashion. But a lot of these pathogens have specific areas that they like to cause infections. And the reason they have specific areas that they're causing infections is because their fimbriae, or we're talking about viruses, their spikes, are allowing them to interact with host receptors in those areas of the body. Okay? And we'll actually talk about this. We might even hopefully get to it next week. But bacteria sex, right, we call that conjugation. And it allows, right, the transfer of DNA between two different bacterial species, but it's not sex in the true sense, right? If we think about procreation, with us, you have half of the genetic contribution from mom, half of it from pa, right? And then you get you. This is just simply copying and then sharing genetic information, but it still is, right, a kind of a crude form of bacterial sex. All right, and we will come back to that. So get excited, right? Some of you never thought that we'd be doing reproductive anatomy and physiology of prokaryotic cells, but here we are, all right? So this is where we'll pick up on Friday, and we'll talk about the internal kind of structure of prokaryotic cells a little bit, and then hopefully we'll get to move into microbial growth, which is actually one of my favorite chapters, because bacteria and archaic and just grow and crazy. But please remember, exam next Friday. Quiz on papers, which I will pull the herd for you there. Make sure you have your two that you want and get your questions. Will be due on, not what you do. We'll take the quiz on Monday, so hopefully you would have read the paper
appointed?
Yeah, yeah.